Male haploid cells through direct spherification. The objective of this study was to design a 3D culture system based on direct spherification to promote neogametogenesis of mouse embryonic stem cells, confirmed by the expression of genetic markers, and utilize these cells to fertilize oocytes. 2D culture systems have been attempted to coax the development of male gametes. However, these systems do not replicate the 3D microenvironment of the seminiferous tubules pictured here. In regenerative medicine, 3D culture systems have been shown to produce functional tissue implants, and recent studies have yielded preliminary success in generating functioning ovarian tissues and testicular organoids. In order to achieve this objective, mouse embryonic stem cells were initially cultured on a fibroblast-coated 6-well plate and later spherified. This video depicts our novel spherification process, in which cells are enveloped by dropping about 50 microliters of mouse embryonic stem cells in media into a sodium alginate solution. This 3D culture system was selected as it better emulates the in vivo environment of neospermatogenesis. The chemistry behind the spherification system is a relatively simple polymerization reaction. The sodium alginate and calcium chloride react instantaneously at 37 degrees Celsius to form a calcium alginate polymer chain, which envelops the cells. Sodium chloride is the sole byproduct of this reaction, making it appropriate for stem cell culture. The resulting spheres, now containing the mouse embryonic stem cells, are characterized by a semi-permeable membrane, allowing for the spheres to be submerged in media. Because of the translucent nature of the spheres, pictures were able to be captured of the cell colonies after spherification. This aspect of our novel process could be vital in wider applications. In order to allow 24/7 visualization under time-lapse monitoring, we resized the microsphere to approximately 200 micrometers using a single embryoscope slide well as a mold. This video shows the colony as it progressively expands and grows exponentially at a rate comparable to a two-dimensional culture system. Spheres of mouse embryonic stem cells were then submerged in Epi-LC media for 3 days, followed by PGCLC media in order to encourage differentiation. This slide lists the active compounds for both the Epi-LC and PGCLC media. After 3 days of exposure to the medium, cells in the two-dimensional culture system achieved only 1% VASA expression. DAZL expression on day 3 reached 29% in the 2D culture. At day 10, both markers were reassessed and maintained comparable expression. Boole was also assessed and found to be expressed at a rate of just 0.5%. The 3D culture achieved 15% expression of VASA and 45% expression of DAZL on day 3. At day 10, expression of these markers remained constant. At this time, Boole was expressed by 10% of cells. Expression of genetic markers DAZL, VASA, Boole, and acrosin were assessed after various points of exposure to the PGCLC media. DAZL signifies differentiation into spermatogonium-like cells and was found to be expressed by 43% of cells on day 3 and 45% of cells on day 10. VASA, which is expressed by spermatocytes, peaked on day 29 with an expression rate of 20%. Boole is expressed by postmeiotic germline cells. Expression of Boole began on day 10, with ultimately peaking on day 15, with 10% expression. Acrosin, a gene expressed in spermatotids, was also first expressed on day 10, at just 2%. Expression peaked on day 22 and 29, with the marker present in 7% of cells. Based on the results, differentiated cells were injected into oocytes after 15, 22, 29, and 36 days of exposure to the PGCLC media. Injected oocytes were monitored in a time-lapse incubator in order to determine the exact time of developmental milestones after injection. Data was collected to determine the percentage of injected oocytes that achieved certain stages of development, most notably the presence of 2 pronuclei, which was considered successful fertilization, and blastocyst formation, which signified complete preimplantation development, were monitored. Control zygotes achieved a fertilization rate of about 88% and a blastocyst rate of about 77%. Oocytes injected on day 15, the shortest exposure time, had a fertilization rate of 35%, 2-cell rate of about 20%, and a blastocyst rate of 5%. Oocytes injected with day 22 cells achieved a fertilization rate of about 61%, 2-cell rate of about 61% as well, and a 17% blastocyst rate. Cells exposed in the spheres for 29 days achieved about 82% fertilization, 70% 2-cell stage, and 36% blastocyst rate. Oocytes injected with day 36 cells achieved a fertilization rate of about 50%, 2-cell rate of about 42%, and a blastocyst rate of about 8%. Our novel 3D culture system, in combination with differentiation medium, successfully induced mouse embryonic stem cells into post-meiotic germ-line cells, confirmed by the expression of the genetic markers DAZL, VASA, BOOL, and acrosin. The putative haploid male cells were capable of fertilizing oocytes and generating embryos with normal preimplantation development. Once the ability to generate live, healthy offspring is confirmed in animal models, and our system is refined in the human model, genotyped gametes for men with germ cell aplasia could be produced to help these men achieve parenthood.